Alrighty, everybody. This is the second to last lecture, I believe, actually. So, we're going to go ahead and take a look at this guy. We're going to be looking at protein digestion and degradation today. And we're talking about, how dietary proteins, how you absorb those, digest them, and then, the proteases involved in that process. So amino acids. So we've talked about amino acids. We've talked about de novo synthesis of the non-essential ones. So like glutamate, glutamine, like alanine. What's it called? Not methionine. Glycine, serine, all that jazz, and cysteine. These are all amino acids you can make pretty well. But there's plenty of amino acids that are required from diet. And we talked about those as the essential amino acids. And, in order to get those amino acids, you have to digest proteins that you eat. So, there's a couple of different proteins, or not couple, there's a couple of different enzymes we have to use, and, we have to digest these things through our stomach and all that jazz. So, they use a bunch of digestive enzymes to, extract these amino acids that are essential to us. So, pepsin, trypsin, chymotrypsin, these are all examples of proteins that we use as enzymes to extract these amino acids. There's also intracellular proteins. So, these dietary proteins are like from our food, right? But with intracellular proteins, there's lysosomes, right? So within a cell, a single cell, the lysosomes able to digest things. There's also for intracellular proteins, there's also ubiquitin and proteasome complexes that you can use. So there's many ways of extracting proteins. So when it comes to the diet, you use these enzymes, but then when it comes to intracellular, you use lysosomes and ubiquitin. So there's a bunch of stuff you can do. So in terms of that, we've got our amino acids, right? So with the amino acids, we talked about it. We can either synthesize proteins or we can degrade proteins back to amino acids. Similarly, we can also use amino acids to make glycolytic intermediates as well as TCA intermediates. And we can also use the glycolytic/TCA intermediates to make amino acids, right? So it goes both ways. So in terms of like our bodies and stuff like that, we generally tend to consume like 70 to 100 grams of protein per day. And we consume it technically as we end up using that much protein to make stuff, if you will. And then we secrete 20 to 30 of the digestive 20 to 30 grams of digestive enzymes per day. Like that's kind of like things that are not useful to us anymore. So these digestive proteases, these things are secreted by the stomach and pancreas. And when we talk about secreted, we don't mean that it's excreted. We don't mean that it's released from the body. We just mean that, during a day's work, in terms of trying to digest our food, it's 20 to 30 grams of these enzymes that are released into our stomach acid every day to digest the food that we eat. So we have digestive proteases. These are secreted by the stomach and the pancreas. They cleave peptide bonds between dietary proteins. And they avoid cleavage of the off-target proteins, so intracellular proteins. So the solution to this is to make inactive forms of these proteins, because you don't want these to be basically, right, the issue with this is like if you just secrete active forms of a so a stomach cell, right, or a pancreatic cell has all these enzymes, right? And it has to make these peptidases before it actually is able to secrete them into the stomach. But the issue is if they're always active, what will happen is, if you translate and transcribe and translate these proteins and they're always active, what will end up happening is that you'll end up actually destroying the cell itself, right? Because these proteases will start going after all of the proteins within the cell. So what you do is you synthesize a version of the protein that is inactive, and then once it enters the stomach's environment, it becomes active. So that's kind of what this is trying to get at. And these inactive forms of proteins are called zymogens. So these are inactive enzymes. They're precursors that require a biochemical change. And in this case, it's usually, I think the stomach acid causes a cleavage of some sort. And so, yeah. here is just a massive list of all of the enzymes. So the zymogen form is literally just taking the active enzyme name and adding -ogen to the end of it. So pepsin, pepsinogen, trypsin, trypsinogen, chymotrypsin, chymotrypsinogen. The only ones that are a bit weird are the ones that end in ACE. If it ends in ACE, we actually put pro in front of it instead. So elastase, we call the zymogen proelastase. For the carboxypeptidase, we call it procarboxypeptidase. So basically, if it ends in syn, it becomes -ogen, and then if it ends in ACE, it becomes pro. And so here you have a kind of the selectivity. So what do they cut? So these are things I would actually maybe put on your note card. I think it's just helpful to know. Pepsinogen and pepsin, I should say in this case, pepsin is selective for leucines and aromatics on the N-terminal side, so on the N-terminus. Trypsin is for basic residues, so lysine and arginine on the C-terminal side. Chymotrypsin for phenylalanine, trityrosine, and tryptophan on the C-terminal side. Elastase for glycine and alanine on the C-terminal side. Carboxypeptidase, just any C-terminal residues, and aminopeptidase is just for any N-terminal residue. So the substrates for each of these, generally it tends to be either the proteins or peptides. And, in this case, it either produces a peptide or it produces amino acids, right? So you can basically see when we go from a protein to a peptide to an amino acid, to individual amino acids. So the main ones to keep in mind are like the first line of defense. So where are they secreted in active locations? So the pepsinogen is secreted by chief cells in the stomach. So that's like your first line of digestion, right? So it should make sense that these guys turn proteins into peptides. Then we actually go on to the small intestine, right? So the small intestine, the pancreas secretes all of these guys into the small intestine. And these are responsible for turning proteins into peptides, right? Except for procarboxypeptidase, which turns into carboxypeptidase. This thing turns peptides into amino acids, but basically, the intestines where the bulk of the digestion happens, I guess I would say. So the stomach is responsible for breaking it down a little bit, so going from proteins to peptides, mostly. And then the small intestine is where there is a little bit of protein to peptide here, but there's also peptide to single amino acids, so it's like a mix of things. So these are secreted by the pancreas into the small intestine, and these are just present in the small intestine normally. So the aminopeptidase is just present normally. So here's kind of like the way that things work. So the stomach, there's a hormone called gastrin. This is released by G cells, which I don't think is very important to know. It's important for the MCAT, but not for this one. But basically, the release of gastrin triggers the release of HCl, which causes a very low pH. And pepsinogen is an enzyme, or a zymogen, sorry, that it gets turned on by low pH. So it becomes pepsin at low pH. After we have, done that, we then have the pancreas release bicarbonate to neutralize all of the acid that's in the stomach. I need to rephrase that. It releases secretin. Secretin is released by the pancreas in response to the low pH, and this secretin triggers the release of bicarbonate by the small intestine. And so once the bicarbonate is released to neutralize this HCl, it also breaks the pepsin, because the pepsin can't be active in basic environments. And then it also activates additional proteases that are active at neutral pH. And so then these amino acids that are broken down are absorbed through the intestine, and they enter the capillaries, and then they get absorbed by other cells. So the main things I would remember here are the main hormones is that gastrin triggers HCl, which leads to low pH, and low pH turns pepsinogen into pepsin. After that, the pancreas releases secretin, and then secretin causes the release of bicarbonate, and then the bicarbonate neutralizes the HCl and stops the pepsin from working. And then once we get back up to around pH 7-ish in the stomach, or in the small intestine, sorry, this activates other proteases like the ones shown here, like these guys up here. And then those allow for more breakdown of amino acids or more breakdown of proteins and peptides. And so these amino acids are then absorbed through intestinal epithelial cells, and they enter the capillaries and then get absorbed by other cells. So here's pepsin, just an image of what pepsin kind of looks like. It's a zymogen secreted by chief cells, stomach, and it responds to pH of 2. And it's a aspartic protease, so basically it uses aspartate in its active form. Basically, the active site has aspartate, if I'm not mistaken. This is usually a pH where most proteins actually tend to denature, but because of how its pepsinogen is actually structured, the quote-unquote denaturation actually leads to a conformational change that activates it. So the low pH causes a conformational change that leads to some autocleavage to form pepsin. And this thing has very limited specificity, though. So it cleaves C-terminals of leucine, phenylalanine, and somewhat of glutamate. So, yeah. So this Sion element here, this segment covers the active site, which is the N-terminal segment. And it's stable at a neutral pH and stabilized by electrostatics. But when we're at low pH, we actually end up cleaving this section off. So the low pH disrupts the binding of that N-terminal segment, and then it's susceptible to proteolysis by itself, or another molecule of pepsin. So basically, it actually self-cleaves itself, or maybe one pepsin will cleave another pepsin, essentially. So pancreatic enzymes, they're stored in secretory vesicles, so a lot of these, along with inhibitor proteins to guard against accidental activation. So they're already in their inactive form, yet generally speaking. But there's also like a double layer of protection, if you will. So in the case of something like trypsin, right, a pancreatic trypsin inhibitor is stored in the pancreas, and it binds in the event of trypsin activation. So just in case one of the trypsinogens in the pancreas becomes active for whatever reason, there's also a second line of defense in the form of these inhibitors within the pancreas that bind and inhibit the trypsin. And once those things bind, they stay bound. Remember that a low KD means a very high affinity. So it pretty much never dissociates. So it kind of like just kills off the enzyme's ability to do anything, if you will. Here is kind of the main thing to keep in mind here in terms of these intestinal ones is enteropeptidase is very important. Actually, it's a serine protease. It's anchored on the outer membrane of these small intestinal epithelial cells. And so once the pancreas releases all its zymogens into the small intestine, the enteropeptidase, which is an active enzyme in the small intestine epithelial cells, or on the membranes of them, basically this thing actually activates the zymogen. Specifically, it activates trypsinogen. So it basically initiates a cascade of zymogen activation, and which yields trypsin, chymotrypsin, elastase, carboxypeptidase, and these all occur at pH 7, which is why the release of bicarb, right, is really important in this whole process in the small intestine. So if we go down here, we see that, this enteropeptidase is in the intestinal cell walls, and so, or the cell membranes, not walls. Mammals don't have cell walls. But yeah, enteropeptidase activates, so once the pancreas releases trypsinogen into the small intestine, the enteropeptidases on the intestinal cell membranes, it activates the trypsinogens, turns them into trypsin, and then from there, enteropeptidase and trypsin ends up self-cleaving itself to make more trypsin. And then from there, all of the other zymogens that were released into the small intestine, like chymotrypsinogen, proelastase, procarboxypeptidase, these are all activated by trypsin. And so they become active once trypsin becomes active, basically. Here you can, we have zymogen activation in the small intestine. We got some examples of this. Here's trypsinogen. Essentially, you have a catalytic triad. Enteropeptidase, what it does is it actually cleaves this section off here to turn it into the active form. So it recognizes like this triple DK sequence, or quadruple DK, sorry. And then it cleaves it between this lysine and this isoleucine here, and then that causes the active form to occur. This is just another mechanism kind of showing where these different enzymes cleave at, basically. Already talked about this, but here's some examples. So enteropeptidase is anchored to the outer membranes of these epithelial cells. Aminopeptidase is also anchored there, and then on top of these enzymes being on the walls of the membranes of these cells, there's also transporters to uptake the amino acids, and these are usually sodium dependent. So basically, it uses the concentration gradient of sodium to kind of move the amino acids into the cell, if I'm not mistaken. Don't quote me on that though. But yeah. Or sorry, sorry. It's not that they use the gradient. It's just that sodium is kind of like a cofactor there. That's probably a better way of thinking about it. Peptide transporters, they use H+. Instead, so like H+ and peptide both move into the cell. So this is basically saying that for the amino acid transporters, the sodium and the amino acid both move in together, and same thing for the peptide transporter, the H+ and the peptide both move into the cell together. Yeah, and then this is just another way of saying that, eventually these amino acids are shot out into the blood, and the blood takes those and sends them to other tissues that need them. This is just a nice diagram of what happens, basically. So you can see here, dietary proteins, we use these enzymes to break them down into this and this. Then the peptides go through aminopeptidase, which is on the intestinal cell membranes, not walls. And then we, transport the amino acids, we transport these peptides. We use our cellular peptidases to turn them into singular amino acids. And then, we use an amino acid transporter to put them in the blood and then shuttle them to other cells that need them. Proteostasis is just homeostasis of proteins. Cool. It just refers to this pathway that cells use to kind of make sure that protein concentrations are relatively stable and stuff like that. So yeah, we'll talk about that. There's protein synthesis, there's protein folding, and then there's protein clearance, because you don't want to just make 8 million proteins, because then you just run out of amino acids, right? If you for example, if you break protein clearance, you'll eventually run out of amino acids. So, and then faulty proteins also cause diseases, right? So you don't want them sticking around for that long. Yeah, there's some other stuff here about why it's important. So in the case of things like glycogen and fats, there's storage forms of them. So glycogen stores glucose, triacylglycerol store fats, but there's no storage form of amino acids or nitrogens. So, and the reason for that is because we need to prevent aggregation of them, right? So remember, a faulty protein can cause disease, right? So you need to degrade them. It's not the glycogen doesn't cause disease, right, and stuff like that. So that's just the reason why we need to make sure that we, don't have a storage for these proteins, because these proteins, as they break over time, they cause disease and stuff like that. So there are intracellular mechanisms of protein degradation. There's ubiquitin pathways, and then there's autophagy, and lysosomal pathways, and stuff like that. So, in terms of in the lysosome is a really cool organelle. It's intracellular, it's membrane enclosed, and it's a digestive organelle. So what it does is it just degrades everything. So it degrades sugars, lipids, proteins, nucleic acids, all of it. The optimal pH activity for a lysosome is a pH 5, and it contains a bunch of hydrolases that are specifically active at acidic pH. So Yeah, it's also important in the control of these enzyme activities and protective mechanisms of the cell. So essentially, what you basically it's a very interesting mechanism, but this here is actually a really cool way of how it's able to actually maintain a low pH. It's just basically using a reverse ATP synthase. So how does it maintain a low pH? And we talked about this. It uses a rotary motor used to pump H+. So it's literally the reverse of ATP synthase. It's a V-type ATPase. So it's an ATP-dependent proton pump, or ATP-dependent lysosomal protein pump. So unlike synthase, which uses the gradient of H+ to make ATP, this consumes ATP to pump H+ into the lysosome. Because the lysosome already has a lot of H+, so it's going against its gradient, so we need to use it. So we need to use ATP. So we use ATP to pump H+ into the lysosome. It's very similar to, the normal ATP with the F0, F1 complexes. And, in the case of the mitochondria, right, we pump H+ into the mitochondrial matrix to drive ADP phosphorylation. But this is not what's going on here. I also don't like the word pump here, because technically it falls down its gradient into the matrix, but you get the idea. So, here's ATP synthase, right? So for ATP synthase, as it falls down the gradient, as it falls down, we make ATP, but in this case, it's the opposite. We're not falling, because falling down should be going this way, right? But we're actually going against the gradient, using ATP and pumping H+ into the lysosome. This is just kind of going over endocytosis a little bit. I don't think this is super important to know. I think it's probably helpful to remember, I guess, or try and have the general idea of what's going on. So, basically, there's endocytosis, phagocytosis, and autophagy. These are cellular pathways that deliver materials to the lysosome for degradation. And so an endosome is basically just like a vesicle. Just think of it like that. But I mean, yeah. So essentially, you can see here we have this protein thingy. This phagosome, right, ends up combining with a lysosome, and then it breaks everything down. In this case, we have endocytosis. This vesicle, like this early endosome, ends up fusing with this lysosome. Basically, it's the same process for all of them, right? For autophagy, we have some sort of phagophore, which forms this autophagosome, and then this lysosome combines with it. Basically, the moral of the story here is that the lysosome just combines with a lot of membranes, and so when it combines with these membranes, it releases its cutting enzymes, and it also releases. Basically, it's a twofold thing, right? It releases its acidic pH into this endosome that it's combining with, and it also releases all of its cleavage enzymes. So you get a twofold thing of denaturing plus cleavage. So that's the main idea there. Now, other than the lysosomes, we also have the ubiquitin proteasome system, which is UPS. It's not the Universal Postal Service. So, yeah. So, a proteasome is a macromolecular machine that is designed to degrade proteins. And it's different from the lysosome because the lysosome is like a membrane-bound organelle, but a UPS is just a protein with some extra complex things involved. So this is the main mean of cellular protein degradation, and it's responsible for 80% of our protein degradation. So it's most of it. So the question is, when do you need to degrade proteins? And the answer to that is, when you're doing regulation, right? So if you don't need to make cholesterol, you can just break HMG-CoA reductase. You can degrade it, right? That's an example of that. So for regulation, it's used quite a bit. So basically, if you want to get rid of a protein, you want to stop a process from happening, you degrade the enzyme that does the process. You can also use it to remove damaged proteins, right? And you can use it to degrade things that are involved in cell cycles. So if you've ever heard of like cyclin-dependent kinases and that kind of stuff, if you don't need those because you're not doing cell cycle, you can just degrade them, right? So that's what you should be thinking about. This is the kind of thing that I unfortunately think they could easily ask you about, like what is the amino acid length of a protease or UPS, of a ubiquitin. Sorry, not a protease, a ubiquitin specifically. But basically, we're going to talk about ubiquitin. So ubiquitin is like a flag. So ubiquitin, UB, is a 76 residue protein that is covalently attached to a target protein. And it's typically bound at a lysine residue. So it's bound at a lysine on the protein that you're intending to degrade. And so the proteasome breaks down proteins that have been tagged with ubiquitin. So basically, you have a protein you want to degrade. You covalently bind ubiquitin to a lysine on that protein. And then that ubiquitin tag allows it to be taken to the proteasome, and you degrade it. There are also other like ubiquitin ligases and deubiquitinating enzymes, which we'll talk about here. So that way you can actually recycle your ubiquitin. Let's go ahead and talk about that. So here we have tagging. There's different types of ubiquitin modifications. You can add one ubiquitin, you can add multiple chains of them, you can all sorts of things, right? You can have, in the case of like polyubiquitin, it's like ubiquitin binding to itself. So there are different types of ubiquitination, and they all have different meanings. So signals a change in, some of them like, signal a change in protein location, some of them change conformation, some of them change activity, some of them change a bunch of stuff, right? Some of them change like vesicles, there's a whole bunch of stuff. But specifically for this class, we're going to mainly talk about proteins that are marked with this, K48 polyubiquitin chains. These are targeted for proteasome degradation. So I would say that these 2 points are not as important as this last one for this class, but that's not to say that you should ignore this. Ubiquitin is not always for degradation, but for the sake of this class, we're going to think about it as mainly this third point. So a polyubiquitin chain here is going to be a case where, we end up targeting these proteins for degradation at the proteasome. And the way that we attach ubiquitin to things is actually through a series of enzymes called E1, E2, and E3. These are all ligases. And this is just this is just what happens. So E1, right, what we do is we take ubiquitin and we activate ubiquitin. So now we've activated ubiquitin. We basically use ATP to attach the ubiquitin onto E1. From here, we use E2, which is a transferase essentially, and we transfer this ubiquitin group onto E2. And then E2 then binds with E3 once, E3 is bound to the substrate. And you get this transesterification reaction where E3 kind of acts like I don't want to say a swinging arm, but it kind of is like a swinging arm, where it basically transfers the ubiquitin from the E2 to E3 and then it to the substrate. And so you end up with this substrate with this lysine, and then, or not substrate with the lysine, sorry, this substrate with the ubiquitin, and then you basically do that process over and over again until you get this big chain. So E1, UB activating enzyme, it's ATP dependent. Activation of the UB at the C terminus, right? That's the C terminus of it. And it basically forms a thioester. E2 is just a carrier enzyme, essentially. It's a conjugated enzyme. It picks up and carries the activated UB to its target protein. And then E3 is the actual transfer enzyme. So once you carried it, the ubiquitin using E3, or E2, sorry, you use the ligase to catalyze the transfer of the ubiquitin from E2 to the target protein, and then it typically attaches the ubiquitin to a lysine residue. And the one thing to keep in mind is this: humans have a lot more E3 ubiquitin ligase than the other guys, than E1 and E2. So like the ratio is like 700 E3s to 40 E2s to 2 E1s. So there's a bunch of E3s, and that should make sense because these E3s are very specific to their substrate. And there's millions of proteins, right? So E3 is specific to the protein, like to a certain subsect of proteins or something like that. And so that's why there's so many E3s, right? E1 and E2, they're pretty general purpose, right? I mean, this is just like a kinase that attaches, or not kinase. This is like a synthetase, if you will. It's like an activating enzyme. That's a pretty general purpose enzyme. This is just a carrier enzyme, E2, just a carrier enzyme. But E3 has to bind a specific protein substrate to bind the ubiquitin to, and so that's a very specific enzyme. And that's why there's so many E3s in comparison to the others. So once we have bound the ubiquitin, now we move on to the proteasome. So how does the proteasome actually degrade the protein when it's tagged with this K48 polyubiquitin? And the proteasome is an efficient intracellular protein degradation machine, and it's found both in the nucleus and the cytoplasm. Take note, it's in both of them, or not just in one. And so what's important about this is that, since you're able to find it in both, you have to be very careful with, what you're able to put in there and what you can't put in there. So all that to say that it's highly regulated, highly processive in nature, and, once you've started the degradation process, you don't want to stop. Because if you release this partially degraded protein into the cell, it's most definitely going to be harmful to the cell. So the proteasome generates peptides that are, about 2 to 15 residues long, and then these can be further, degraded by cellular peptidases. So, yeah. The proteasome, this is the structure of it, basically. So we've got a couple different regions. We've got a 19S regulatory particle and a 20S core particle. You don't have to worry about S. What S means here, it's a measure of density, sedimentation time. Not important for the class. What you should know is, in the current case of the 19S subunit is going to be these two sides, and the 20S core is going to be everything here in the middle. So this is where all the enzymatic reactions occur to break things apart, and the 19S regions are where we do the process of essentially identifying, is this protein bound to ubiquitin or not? So, for the 19S regulatory subunit, what we do is we have ubiquitin first has to interact with a motif that recognizes this polyubiquitin tag. So the 19S subunit has like this binding site for this polyubiquitin thing. And it's basically a thing that controls access to the beta subunits, right? It basically like stops the beta subunits from just eating anything. So once ubiquitin binds with its protein that is attached to, the enzyme deubiquitinase cleaves off the ubiquitin chain, and that ubiquitin is recycled. And then we use an ATPase, so in the beta subunits basically, or not beta subunits, but then we use an ATPase to unfold this target protein and kind of like feed it into the proteolytic core. So basically like we're untangling it and then feeding it one by one into like amino acid by amino acid into the core. So it's kind of just to recap that, the first thing we have to do is recognize the polyubiquitin tag. Then we have to deubiquitinate it, so remove the ubiquitin, recycle it. And then we have to take the protein that we just deubiquitinated and feed it into the core of the proteasome, and we do that through an ATPase. So the ATP is hydrolyzed, that energy is used to break the intermolecular forces, within the protein. And then, when we break those intermolecular forces, we feed it into our proteasome, and then from there, we chop it up into individual amino acids in the core. So the alpha subunit in the 20S core is what controls access to the beta subunit. And so the beta subunit is just a threonine protease. It cleaves the C-terminal side of just any residue. It does not matter, pretty much. But the beta subunit has, or so the alpha subunit has a couple conformations. It's got a closed conformation and an open conformation. This is the beta subunit, sorry. But the alpha subunit has a closed and open conformation, and the way that you kind of open it is through like this stuff up here, right? So, you know, if you ubiquitin binds to the 19S, right, and then it gets deubiquitinated, and then you use ATP to unfold it, then the alpha subunit will start to open up. And then once you get into the beta subunit just chews through everything. So it cleaves the C-terminal side of acidic, basic, or hydrophobic. Just cleaves pretty much everything. And here is a nice image, actually, of what is happening. So I think this is a great image. Just to remember that, you know, this structure is actually on both sides, right? So you can see how it's, like, symmetrical. Right? I think that's something to remember. It's people kind of forget that, but try and remember the symmetry. Yeah, so, I mean, you know, we talked about the 19S, right? So there's, you know, the ubiquitin binding site here on this side. And then you have the ATPase ring, this ring here, which is, you know, still part of the 19S subunit. And then you have the alpha region, which is kind of like the door. And then the beta subunit, which is the actual cutting portion. But other than that, that's actually going to be the end of this video. I hope it was helpful. Bit on the longer side in terms of the recent videos today, but only a couple minutes more, so it's not like a crazy longer video. So yeah, but other than that, I hope this video was helpful. Gonna have a workshop on Tuesday for you, before your guys' exam on Wednesday, I believe. So feel free to pop by for that if you would like, ask questions, all that jazz. But yeah, other than that, happy studying.